Hello. Welcome, everyone. Thank you guys all for coming to the last of this summer's Wednesday at the Evening Genome Talk. I'm really excited that we get to close out this talk with Dr. Abitua, Phil, who's one of the assistant professors here in the Department of Genome Sciences. You guys will see some absolutely beautiful images and some really cool data over the next hour, because I know every single time I see a talk by Phil, I'm always leave completely amazed. So I'm really excited to see him talk tonight. Just for background, so Phil did his undergraduate at UT Austin, went over to Berkeley where he did his graduate studies, and at that time really was focusing in on how does evolution, and specifically evolution of really new or cool features of the body plan, happen in different organisms. And really, I think one of the things which I love the most about his research is he's not necessarily just focused on sort of some of the tried and true model organisms we look at, but really trying to push the boundaries in terms of understanding how has biology done this in really cool and novel ways. This has resulted in, as you can imagine, many high-profile studies in the literature, including one that just came out a couple of weeks ago in the journal Science. And I'm really looking forward to hearing more about what your research is today. So thank you very much, Phil, and looking forward to it.
All right. Hello, everyone. I'm Phil Abitua. I'm going to try something a little different today. Normally, I would give a research talk, but I was given the task of giving a talk to a general audience. And so I'm going to attempt to sort of explain some of the concepts of developmental biology, some of the questions that a developmental biologist might ask. And I'm going to focus a little bit more on the evolutionary origins of animals. And so I would say the majority of this work is not my own, and it's somewhere, I guess, between a story of my own career. There's a little bit of my own work at the end, and maybe a sort of 101 lecture of what development is and how people end up studying it. If any of this is unclear, like, please stop and ask me questions. I would like to keep this like as casual as possible. So my first foray into developmental biology was at UT Austin with John Wallingford. And during the first day of class, John showed these beautiful time-lapse movies to all of us. And what you're seeing here is the process of neural tube closure in a salamander. So you can see that this region here is the flat neural plate, and that ends up folding into a tube, and that tube ends up giving rise to your spinal cord, as well as your brain in this anterior region. And this beautiful process is very highly conserved through all sort of vertebrate embryos. And I think I was really just inspired and struck by the beauty of these images. And that really sort of inspired me to learn more. And so during the first lecture, I asked John if he had places in his lab for an undergraduate to do research. And I was kind of amazed that he did, and he gave me that opportunity. And that was the beginning of my sort of scientific journey. So what is it that a developmental biologist studies? This is basically how a single fertilized egg ends up giving rise to a complex body plan. And so you can see here that there are different left and right asymmetries. We have a very complex nervous system, as well as a system of organs. We have skin and guts and bones, and this amazing diversity of tissues that all works together in sort of unison to produce us. And developmental biologists, their sort of goal is to understand this process at a very mechanistic level. So they're interested in what genes are turning on, the patterns of those genes, what genes are turning off, what proteins are being made, how the embryo is taking shape. So that there are many sort of complexities sort of unfolding very reliably in every single embryo. So that's kind of what in essence a developmental biologist is seeking to understand. And another question that I became interested in sort of later during my career is how all of this evolved. And so, obviously, as humans, we exist as vertebrates, but there are many different forms in the animal kingdom that produce many different cell types and shapes and have different specialities. And I think it's really just kind of astonishing to think about the number of forms and complexities that exist in the natural world. And, I think in a way, being a developmental biologist is a very sort of easy concept to grasp, because I think it really is part of the human experience, right? It describes where we come from, right? And so I would say, I would say that most of you in the audience have been concerned or thought about the development of a child and either their family member or some loved one, right? And so I think we've all sort of pondered this question about where it is that we come from and how this occurs. So I'm going to sort of start at the beginning of what the first sort of animals were like. And so let's see. For most of time, right, for approximately 4 billion years, all of life on the planet existed as single-celled organisms. And these organisms were found in these deep sea vents in this gassy metal-laden area of intensely hot plumes that were caused by the salt water interacting with magma at the ocean floor. So there were these very intense environments where the first single-cell organisms began to take shape. And like I said, single-celled organisms dominated the planet for most of the existence of Earth. And then something really miraculous happened around 600 million years. And just to give you some context of what the planet looked like 600 million years ago, there was one large supercontinent, and that was known as Pannotia. And this is the period when multicellular organisms first emerged. So this was kind of before Pangea, again, 600 million years ago. And so one thing that we do as developmental biologists, and especially those that are interested in the evolutionary origins, is important to understand the context of how all of these creatures are related. And so what I'm showing you here on the screen is a kind of graph called a phylogenetic tree, which gives us an understanding of the evolutionary relationships of different organisms. So up here in this bracket are all of the sort of multicellular animals that exist, the sort of major phyla. So that would include ctenophores and things like sponges, and then these sort of jellyfish-like creatures, and then most other animals that you would recognize as animals, vertebrates, that includes mammals, and then also insects and crustaceans and those types of creatures. And if we want to study how multicellularity evolved, one common thing that people like us think about is the last common ancestor. So outside of this is a group of single-celled organisms known as choanoflagellates. And the great thing about choanoflagellates is that they exist on the planet today. They're an extinct they're an existing species that we can study in the lab. So if we're interested in the origin of multicellular animals, what we can do is use this group as a proxy for the last common ancestor. So the ancestor that gave rise to all of animals, as well as this single-cell choanoflagellate. So let's look at what a choanoflagellate looks like. So this is an image of a choanoflagellate. They have a flagellum that they stick out into the sort of water, and with that flagella, they're generating a fluid flow, and that's attracting bacteria to this collar structure here. And one thing that I think people are really, who study choanoflagellates really find quite interesting is that they're able to form a rosette structure. So in order to facilitate feeding, they go through a life cycle where they make these little aggregates of multiple different single cells, and they work together to generate a fluid flow to capture bacteria. And this is known as this rosette. One thing that I think is just really almost astonishing and kind of unbelievable is that in a sponge, which is a multicellular organism, in the cavity of the sponge, there are cell types known as they're called choanocytes, and these line the sort of cavity and facilitate in feeding, and they look remarkably similar to these choanoflagellates. I mean, I think it's quite astonishing, the similarities between the appearance of this organism and the cells that line the sponge. And so it turns out that back in 1886, Eli Mershchenkopf, he's this Russian developmental biologist who came up with this theory that choanoflagellates were the relative or cousins to all existing animals. This is before this type of imaging existed, which is pretty amazing. And then 122 years later, a pair of researchers at UC Berkeley, Nicole King and Dan Roxar, were able to prove using genetics and genomic technology that choanoflagellates are indeed the ancestor to all common multicellular animals. So, that's kind of like the types of questions a developmental evolutionary biologist is interested in asking, and we've been contemplating these questions for hundreds of years. And really only in the genomic era are we able to come up with these, or are we able to validate these hypotheses? All right, so I talked about this rosette structure that choanoflagellates can form. Does anyone have any ideas as to how this structure forms? How would you end up getting this rosette? Any ideas? By just sort of size, but that's and that's a good guess about the geometry. Does anyone have any just simple ideas of how this forms? Yeah. Yes, that is important. The orange part being sticky is definitely a critical part. Anyway, so there's kind of like two very broad ways in which this sort of multicellular structure can form, and my son seems to have some ideas, and that would be aggregation or cell division. And for a long time, those were the two main competing theories. And aggregative multicellularity has evolved in eukaryotes and in some bacteria. I think Isaac might need to head out. So this sort of aggregative multicellularity has evolved many times in the past, and it's seen to be largely kind of an emergency response where they're sort of low on resources, or that they need to disperse themselves, and this is indeed something that we can see in the animal kingdom. So how many of you have heard of slime molds before? Slime molds. All right, most of you have heard of slime molds. Some slime molds exist as sort of a single cell state. They can aggregate together, and then through cooperation, they're able to make this stalk. So it sort of elevates their sort of structure and allows for them to disperse spores. So people thought that this really could have been a way in which multicellularity evolved. And then I think the maybe more simple explanation is through cell division, which is how this occurs in choanoflagellates. All right. So the idea is that this might make them resistant to predators. It may facilitate that cooperative feeding. And I think the most important thing that multicellularity allowed was this concept of a division of labor. So as was suggested by one of the audience members, cell division and this pink stuff called the extracellular matrix, it was what were really like the key sort of steps required for multicellularity. Many of you have probably, maybe some of you don't know what extracellular matrix is, but I'm sure many of you have heard of collagen, which is a fibrous kind of material, which is part of the extracellular matrix, which is this pink glue that kind of holds everything together. All right, so again, now you have cells, they're dividing, they're able to sort of stick together. And this allows for this division of labor, which allows cells to become specialists. And this really is one of the major innovations of animals, right? Is that cells of an organism can do different things. And so fast forward about 100 million years later, so 500 million years ago, Pannotia is beginning to break up. The sort of land is very barren. There is no vegetation, and subsequently there really is no soil, and so it's really like a barren wasteland on the sort of landmass. But deep in the ocean, there's some really exciting things happening, and I'm sure many of you have heard of the Cambrian explosion. And during this condensed period of time, most of the major animal phyla that, the main groups of animals that we sort of recognize all emerged during this same period. And this would include vertebrates. So, this really was like an explosion in the terms of the amount of diversity that occurred during this time period. All right, so a little more on cell types. So the first two cell types of the embryo are the ectoderm and the endoderm. And the ectoderm is essentially an outside layer, and the endoderm is an inner layer. And these two major groups of cells are called germ layers, and they give rise to all of the tissues and organs of animals. And the outer layer is for protection, as well as sensing. And then the inner layer is primarily for digestion. And your ectoderm produces skin and nerves, whereas the endoderm produces the gut. And there are animals that are relatively simple that have only these two germ layers. They have basically inside cells and outside cells. And these include ctenophores, which maybe some of you may have seen at an aquarium, as well as cnidarians, and those include like hydra, jellyfish, sea anemones. These are relatively simple in their body plan. They're mainly radial, so they don't really have like a left or a right, and they're really composed of these two simple cell types. So one question that developmental biologists commonly think about is how an embryo takes shape. And so you might be wondering, how did the endoderm get inside? How does that occur? And this is really the first shape change that most embryos undergo. And so here's this hydra. You can see that it has this adult body plan. But early on, it is essentially just a ball of cells, and some of those cells end up internalizing into the embryo. So you can kind of imagine it's like you have a balloon and you're like sticking your fist through the balloon, and cells are kind of moving inside. And so this is this process here. This movement of cells of the endoderm in pink going inside is the process of gastrulation. And so cells or the embryo can take shape through many different ways. There's cell division, there's cell movements. The individual cells themselves can change shape. Cells can die. There are many ways in which this occurs, and it in itself is kind of a whole field of developmental biology. But as you can see here, the endoderm is sort of already highlighted as a special, unique part of the embryo that's different from the ectoderm or the outside. And so the process of the endoderm becoming endoderm or becoming different from the ectoderm is what is known as differentiation. And I'm sure I see many of you have degrees, but I will explain this in sort of more simple terms, kind of how this difference unfolds. So this is kind of like developmental biology 101, or sort of like just general biology. But inside of every single cell of an organism, there exists the same DNA. So the blueprint that is the instruction manual for making any given organism is the same in every single cell. And then this ends up being read out into a different three-letter code called RNA, and that is different in every single cell. And this RNA becomes protein, which again is different in every single cell. So this is sort of the central dogma of biology, and explains how the differences in our body emerge. And there is a special type of protein, and that protein is called a transcription factor. And these are essentially molecular switches, which have the ability to bind the DNA and then make cells and then produce RNA differently in each of our cells. This way, we end up, growing an arm or a brain in the right location. And so, if we go back to this endoderm and ectoderm idea, the endoderm has one set of molecular switches, and the ectoderm has a different set of molecular switches. And these are very sort of centrally important to what developmental biology is. And through this, we can actually end up seeing like where these switches are coming on in the embryo. So again, this is a Hydra embryo, and we can see, we can actually visualize where the RNA is. And so in black, you can see there's one molecular switch called snail turning on in the endoderm, and then the ectoderm is marked by a different molecular switch. And this technique of looking at the RNA in embryos is really like kind of a bread and butter technique of developmental biology. And the details aren't very important, but the genes of a developing embryo turn on in a myriad of like very sort of beautiful patterns. You get sort of polka dots, right? You also have genes sort of turning on in stripes. You also have, genes being expressed in particular regions of the embryo, such as the sort of hands and feet here. And we can also look at where genes are being expressed with kind of multiple probes. We can look at multiple genes turning on in a single embryo. So here's a fly embryo, and I believe there's 5 different genes labeled here in this single embryo. And these are the types of patterns that developmental biologists really, I think, love to sort of examine and decipher where these different genes are turning on. And I think, in a large part, they're kind of very beautiful and attractive, just the sort of myriad of expressions that genes can exhibit. All right, so this is like kind of the last part of the sort of conceptual part of what developmental biology is. So integrating this all together, in the very beginning, you have a fertilized egg. Those cells continue to divide, and at this point, like all of the cells basically express the same genes, and then different molecular switches are turning on in different patterns, right? And when that occurs, those cells can start behaving or moving around or making special proteins to contribute to different cell types. And this process just basically continues throughout the duration of development, which in some animals can last for like a day and others for months, right? So this is basically what developmental biology is. And in these very simple creatures, I think it's interesting to think about like how many cell types do you need to make a simple organism? And this was something that we've been asking for a long time, but only recently in kind of the modern era have we had technology that sort of really changes the game and allows us to investigate this a little more carefully, or like sort of, I guess, in a very complete way. And now what we can do is essentially take a whole embryo and dissociate all of its cells, kind of break them up into single bits, and we can put them in this kind of magic toaster device, and then we can sequence the RNA of every cell. So not only are we looking at 1 to 2 RNAs, but we're looking at hundreds of RNAs through a method called single cell sequencing. And this is a representation of all of those cells. So each dot is a cell, and then they're clustered by how similar they are. So in a hydra, in this relatively simple creature, there are approximately 8 different cell types. And again, this is kind of a pretty simple creature, but in humans or vertebrates, we have many more cell types. Does anyone have any guess, like, as to how many cell types like a mouse would have, or a vertebrate, if a hydra has 8? I don't know. At least 9. 50. 50 is a pretty good guess. That's a pretty convoluted question, because what is a cell type, I think, is something that's heavily debated. But there's something on there's approximately about 100 different cell types. And so work by members of the Genome Sciences Department, Cole Trapnell and Jason Jury, basically used this technology to look at this process of cells becoming different, differentiation in a mouse embryo. And this is kind of just like a subway map sort of visualization of this process. But basically, at the 1 cell stage, you start here, and then you can take different routes to become the skin, or the gut, or blood cells, or muscle, or limbs. And all of these little names here as you go along are those molecular switches turning on and turning off genes. So this is just to show you that we're kind of at this point where we can collect a tremendous amount of data, but in all honesty, it's very hard for us to fully understand what is happening still. I mean, we can kind of see the sequence of events, but we're very far from understanding the complete story of how animals develop. And so one thing that I personally am very interested in thinking about is, where did all of this diversity come from? And I was interested in this sort of area of the map here. And so I'm going to go back in time again to a little more of a modern time. Berkeley, in 2009, there was this great recession, a record unemployment. I was pretty happy about my decision to do a PhD at Berkeley and avoid getting a real job. But maybe some of you, I know some of you have PhDs, but maybe some of you are wondering, like, what is it like to get a PhD, or what is a PhD? So we can think of this circle as all of human knowledge. And when you go to elementary school, at the end, you know something, right? And then by the time you finish high school, you know a little more. And then when you go get a bachelor's degree, you start specializing in some field of research. So that may be economics or writing or biology, right? So you're starting to develop some speciality. And then when you get a master's or a PhD, you're really trying to intensely learn about a particular subject, and you're sort of pushing the edge of human knowledge. And you'll continue pushing on that boundary for several years, right? And until one day that boundary gives a little bit. And that is essentially what a PhD is. And so when you get a PhD, after all those years very, very intensely doing research and reading, your world looks very different to you, right? You're kind of off in your own headspace. But it's important to remember the bigger picture. And I know many of you have PhDs or pursuing PhDs, but I encourage all of you to sort of continue learning and sort of continue pushing the bounds of human knowledge. All right, I'm going to spend a little bit of time talking about my own research. So at Berkeley, I studied with this guy, Mike Levine, who was important for understanding, I guess he's most famous for kind of understanding what those molecular switches were kind of back in the 80s and the 90s. And I was interested in cell types that were unique to vertebrates. And particularly, I was interested in the origin of neural crest and placodes. And neural crests are an amazing cell type. They produce most of your cranial skull vault, as well as the majority of the pigment in your body, all of your peripheral nervous system, and the cranial facial cartilage of your head, so your ears and your nose. And then there's another cell type, placodes, which produce the sensory structures of our ears and of our nose. And so these are very important for it's believed for the success of all vertebrates, right? I mean, it kind of puts you at the top of the food chain if you have a jaw and you can, really sense your environment very well. So I'm going to tell you a little bit about the embryonic origins of neural crest. Essentially, if you think back to that movie that I showed you in the very beginning, neural crest develop from the area where your spinal cord is developing. And the neural crest cells in your head, again, produce the skull vault and your cranial cartilage and part of your heart. And one really beautiful thing about neural crest is that it has this ability to produce these cells that migrate out into streams of your sort of extremities. So, your nerves are all over your body, but they originate from your spine, and so they have to migrate throughout the body. And back in 1983, these two gentlemen, Gans and Northcutt, came up with this sort of really influential work that said that cranial neural crests were clearly the most striking change coincident with the vertebrate origins. So they basically were saying, like, this cell type made us uniquely who we are. So as I mentioned, all vertebrates have neural crests, so sharks, fish, frogs, reptiles, mammals. And in order to understand where these cell types came from, we have to then look at the sort of related organisms, and those include Amphioxus and tunicates, or cephalochordates, Amphioxus, lancelets. They have many different names. So has anyone heard of a cephalochordate? Cephalochordate, okay. Well, here's a picture of a cephalochordate. For the longest time, people thought that this was the relative to the vertebrates. You can see why they might think that. It does have a very sort of, for lack of a better word, like a fish type appearance. I don't know, to me, it kind of just looks like a piece of sashimi, like stuck in the sand or something like that. But alas, these organisms do not have neural crest-like cells. And what about tunicates? They're also related to vertebrates. Here's a picture of tunicates. I don't know, to me, and I think to most people, you would be surprised to think that these are actually very closely related to fish. And that's because they undergo a very dramatic change during their development. So, but if you look at the juvenile stage, they have this swimming tadpole sort of body plan, and they undergo a metamorphosis like a butterfly. So they change they develop very differently in their adult form. All right. So in order to understand where neural crests come from, we have to, understand what homology is. And so I think many of you can appreciate that a human arm and a cat arm have some similarity. And the same thing can be even observed in a whale or a bat. They're just kind of different extreme versions of that. So you can see that these are the digits here in blue. And this is not just a similarity, but this is actually homology. So these structures are similar because they come from they share a common ancestry. So here's that on the screen here. And in a similar way, since these structures in our body are simply composed of cells, we can ask, how are cells related? Just like just like we can think about how arms are related or how animals are related. And so I was interested in looking for homologs to these neural crest cells and tunicates. So here's a picture of a tunicate. And then kind of in this region, I found cells that were sort of in that location. They both make pigmented cells, and they express some of the same molecular switches. But importantly, they don't express all of the same genes. And these cells in tunicates don't migrate. And I was sort of naively wondering, like, what would it take? What would it take to evolve neural crest cells, knowing what we know about these cells in tunicates? So I basically looked for genes that tunicates did not express, but that the vertebrates had. And I introduced those neural crest vertebrate genes into these cells. And essentially, I'll just go through this really quickly, but here are those neural crest-like cells. And when you misexpress, or when you express this molecular switch called twist, all of a sudden these cells start spreading over the body. And you can follow these later in development. Normally they're located here, but upon the expression of this molecular switch, you're activating that migratory program and allowing these cells to move across the body like they do in vertebrate neural crest cells. So we came up with this hypothesis that in the ancestral organism, there was an ancestral cell, and in the lineage leading up to vertebrates, there was the expression of twist, which was important for making cranial neural crest. And you know, I was thrilled beyond belief that we were able to publish this in Nature. I got this image off of Wikipedia, surprisingly, and photo credit to Nick Hopgood. And we did similar work with placodes as well. I'm gonna zoom through this so we don't run out of time. Now I work on, when I went to do my postdoc, I started working on the African turquoise killifish, which is the organism that I'm currently studying. And one thing that I think is really interesting, switching gears here, about these fish is that they live in these extreme environments. So African turquoise killifish live in these ephemeral ponds. So during the dry season, the water completely evaporates. And as you might, you might surmise that it's very difficult for a fish to live without water. And the way they are able to survive this extreme condition is that the embryonic development of these animals can basically pause. And it can pause for periods of long as 6 months and possibly even up to a year or longer. And so I think this system is really interesting because they live in this extreme environment. So the selection that is on these organisms might produce kind of very novel features that are unique to these organisms. And hopefully we can understand, this process of evolution by looking at animals that live in this very unique environment. So these animals, again, have interesting characteristics. They're able to pause their development. They're very hardy since they live in this sort of muddy substrate. They're very resistant to UV damage. They also have cell types that other related organisms do not possess. And they also have a very unusual way of developing, so that early morphogenetic shape that the animal undergoes is highly unusual compared to other vertebrates that people have studied. And so, by studying these outliers, these creatures with extreme adaptations, we can learn sort of about the extent to which evolution can change the biology of organisms. So just at the very end, I'll go back to this question about how does this initial asymmetry develop? How does the endoderm become different from the ectoderm? And well, you might wonder how this is all set up in the first place. And it turns out that in most fish and frogs, there's a pattern of mRNA that is adopted from the mother. So the mother lays some genetic information here that ends up being inherited to one side of the animal. And this is known, this is like a pattern that exists before this gene expression first emerges. And when we look across animals, we see that most fish and frogs develop in this way, but mammals somehow miraculously have this initial pattern emerge in a way that's self-organized. And people don't really understand precisely how this works. I just want to stress that, this is basically like the most simple question in developmental biology, like how does the first pattern emerge? How is the symmetry of the animal broken? And even after studying embryos for hundreds of years, this very simple question is not fully solved. And it turns out that the killifish, since their development is so unusual, these pre-pattern mRNAs, they just like don't exist. They've actually lost their function. And we published recently that the killifish ends up developing without this pre-pattern, similar to mammals. And so, I don't know, I just want to end on the note of like, biology is very complicated and we struggle to understand the most basic questions still. And it may seem from like reading the textbooks that we have everything figured out. But I think that's, very, very far from the truth. And it's a very exciting time to be a developmental biologist, because all of these amazing tools have really emerged in my very short career. And so, I don't know, it's really an honor to, be studying animals. It's a lot of fun. Okay, and with that, I'll be quiet. Thank you to my lab for just being very supportive, as well as my family, who unfortunately had to leave, I guess. But yeah, I'm happy to take any questions, although I guess there is not time for questions, but I'm happy to stay around for a little bit. Apologies for going over. All right, thank you.
Questions? If I can get started. These organisms that only have ectoderm and endoderm,
Yeah.
they've evolved for millions and millions of years. Why don't you think they've evolved something in addition to that?
Yeah. So some of them have. Yeah, that's an important thing to remember, is that after animals split in their lineages, they continue to evolve themselves, and so some of them will lose features, and some of them will gain sort of kind of unique traits. But one idea, at least for vertebrates, is that genome duplication allowed for the sort of raw genetic material to produce new cell types. So, like I said, the vertebrates all have neural crests, and it's believed that this is a consequence of their genomes expanding rapidly. So, there's some correlation with like the complexity of the genome with the complexity of the organisms, although the absolute size certainly does not scale in that way, but yeah.
Could you say a little bit more about the pigment thing in neural crest cells? I didn't really follow what the relationship between those two things are.
Oh, yeah. So all of our pigment in our body, and same thing with fish, come from neural crest cells. And the tunicate, the cells that we believe are homologous to neural crest, also have the ability to produce pigment cells. So there's one very specific transcription factor that is the master regulator of making pigmented cells. And so you can imagine, it's hard to understand the sequence of events. Like what migrated first? What evolved first? Was it the ability to produce pigmented cells? Was it their migration? Which of the many derivatives, the neurons or sort of cartilage or bone, the sequence of events of what came first is hard to understand, but at least we can say that most likely in the last common ancestor, there were cells along the sort of edge of the spinal cord that could produce pigment. That wasn't really what was the like missing link, if that makes sense. That at the neural tube border, there were already cells that could produce pigment, and that predated vertebrate neural crest.
I'm curious about how these neural crest-like cells in tunicates are poised to become migratory when you misexpress twist. And did you ever see that just misexpressing twist caused other cells to become migratory, or how does that work?
Yeah. So that is a great question, and so that is interesting. So we tried misexpressing twists in other parts of the body to see if we also would gain that migratory ability, and it turns out that was not the case. So yeah, we express twist at different times and in different tissues. And it seems that along that neural plate where the spine is developing, since cells are already kind of moving, they express genes that allow those cells to kind of, I guess, be less adherent than cells of the ectoderm, essentially. So it's believed that in order for a cell to migrate, it needs the dual expression of twist and snail. And it turns out that at the neural plate border, snail was already sort of present. So there were switches that allowed the embryo to take shape, and maybe you needed just a little more additional factors to allow cells to kind of wiggle free is kind of the way that we thought about it. That's a very good question.
From Zoom, Susan McNabb asks, you said the killifish have unusual morphogenesis. Is development patterned, but with the pattern emerging later than most embryos, or is it unpatterned?
Yes, so we believe that there really is no pattern initially. We can say pretty confidently that the gene that sets up that initial pattern in other frogs and fish is a pseudogene, so it has lost its function. There's a frame shift mutation. Could there be other patterns that people don't understand or have not described yet that sort of tip the scale in order to initiate patterning? That certainly is a possibility. there's some really interesting stuff where the killifish just normally, when they develop, occasionally not a single embryo will form, but you'll have multiple axes emerge at the same time. So in sort of a twinning effect. So we do believe that the pattern emerges later, and this may be due to the fact that the embryos can interdiapause, so they can exist unpatterned for a very long time until the conditions of the environment are, good for continuing to develop. Good question.
Are there parallels in the plant world in the evolution of multicellularity? Because I think there are algae whose names they don't remember, like 4-cell green algae and 16-cell algae, where there starts to be some sort of specialization, very rudimentary forms.
Yeah. So, that is not my area of specialty, but from what I understand, like multicellularity across, archaea and bacteria and plants, that evolves independently multiple times. And as far as I understand from my reading of the literature, animal multicellularity that gave rise to all of the creatures as we know as animals really only evolved once. So yeah, I mean, I don't know the exact details of that, but certainly.
Because I have another question. You talked clearly about the utility of the neural crest and the placodes for innovation
Yeah.
within the body plan. Have the other germ layers just maxed out under innovative capacity, or do you think there still is innovation within, mesoderm, ectoderm, and endoderm?
Yeah, that's a great question. So in that sort of subway map, you can see that it's kind of like a ever-branching fractal of complexity, right? That at the edges, there's just more and more branches. And the thinking is that like those terminal nodes are more likely to still have the capacity to evolve. And that's kind of intuitive because like all of development is sort of a hierarchy, right? And so, you're less likely to get very early changes in development, like the addition of a new germ layer, because that could have huge consequences to the rest of the sort of development of the different cell types that come after it. And yeah, funnily enough, like some people refer to neural crest as the fourth germ layer because it is so unusual and really has properties that are sort of the hybrid between the mesoderm and the ectoderm. But I think the answer to that question is, I think there's plenty of room for evolution, but more at those terminal nodes than at those very early steps. But still, under extreme pressures like the annual killifish, you can change the way that program sort of emerges. I was like trying to think about what I could share with everyone, and I was like, there's way too much. Like I could come up with like a whole lecture series to explain development. I guess that would just be a class. So but anyways, it's a super fascinating question. All right. Well, thank you all for coming and listening to me ramble.